Thank you, Ed. Thank you. So thank you, Ed, for the opportunity to come and speak this afternoon, and thank you to all of you for sweltering through the heat to come here and sit in a relatively cool lecture theatre. At least you can nod off in peace and quiet as I entertain you for the next half an hour or so. So it's my very great pleasure to be able to introduce you to, perhaps, you've heard a lot about insects today, perhaps this is the best, the most charismatic of insects in Britain, if not the whole world, the burying beetle, or sexton beetle, as it is sometimes called. So has anyone here had the privilege of seeing one of these beetles in the wild? Yes. Anyone else familiar with it? Brilliant. So you are in good company, because the wonders of the burying beetle were first made famous to the wider world by the great French naturalist Jean-Henri Fabre a little over 100 years ago in his book, The Wonders of Instinct. And so here's the great man himself over on the left, looking drained by the sheer magnificence of his own prose. Because here is what he had to say about the burying beetle. It's the most vigorous and famous of these expurgators of the soil is the burying beetle, so different from the cadaveric mob in dress and habits. For he is a gravedigger, a sexton. He buries a corpse entire on the spot in the cellar, where the thing, duly ripened, will form the diet of his larvae. He buries it in order to establish his progeny therein. He makes a clearance of death on behalf of life. So that's the peak of the entire talk. It's downhill all the way now. But in much more basic terms than Fabre was describing, here is the life cycle of the burying beetle. So round about now, in fact, this evening probably, in the woods around Cambridge, burying beetles will be actively searching out the newly dead as a breeding opportunity. So they fly around looking for a small mouse or a songbird, and if they get lucky, they find one, and if they get really lucky, then a partner will show up too. And together, they'll set about converting this dead body into a nest for their larvae. So they strip off the fur and feathers, they roll the flesh into a ball, and they bury it in a shallow grave. And all the time they're doing this, they're smearing the flesh in fluids from their mouth and from their anus. And while they're busy making the nest, the female lays her eggs in the soil surrounding the carcass. So this takes about 3 days. And then the larvae start to hatch and they crawl through the soil to the nest. And so we're going to switch to some home movie of burying beetle family life to see what happens next. And what happens next is shown here. So this is the nest. This is a mouse in a former life. Here are the larvae all snuggled up inside. And you can see that they busy themselves by munching away on the dead body to feed themselves. But from time to time, a parent will show up. There we go, on cue. Will seek out individual larvae and will transfer fluids directly from their mouthparts to the mouthparts of a single larva. And because of remarkable acts of care like this, which are undertaken by both parents, the burying beetle is a rare example of an insect that provides biparental care. And so what that means is that the larvae are defended by parents during this very vulnerable stage in their lives, because the parents are hanging around to look after them. And because they're being fed, they grow very fast. So within a week, there's nothing left of the dead mouse except for a few bones. And the larvae at that point start to crawl away into the soil, and the parents fly off in search of new reproductive opportunities. And in the soil, the larvae will pupate. That takes a couple of weeks. And then they will emerge as a sexually immature adult. And then after another 2 to 3 weeks, they'll reach sexual maturity, and the whole life cycle will start again. So the whole thing takes about 7 weeks in total. And amazingly, we can stage the whole life cycle in our lab. So I just want to quickly show you the setup in our lab so that when I go through the experiments that I'm going to tell you about this afternoon, you can imagine how we do it. So this is one of our study sites around Cambridge. This is Wicken Fen, where we trap beetles. We bring them back to the lab. Each beetle gets its own personal little box with a unique identity label. When we want to breed them, we put them in a larger plastic tub like this, lined with a bit of soil, furnished with the dead mouse. We pop in a pair put the lid on, put them in a cupboard to simulate underground conditions, and then we come back a week or so later and the breeding cycle's complete and we can count and weigh the number of larvae that are produced. So with this setup, we can manipulate the details of the environment during each breeding event, and that allows us to carefully understand what goes on each time a pair of beetle breeds and the kinds of challenges they have to overcome when breeding on a dead body. Now, one major challenge that beetles face is the level of competition for the dead body itself. And to begin to understand the scale of that challenge, we're going to look now at what happens to a dead mouse in the hours after death if it's not lucky enough to be found by burying beetles. And it turns out there's a lot of time-lapse movies like this on YouTube, so I've just stolen it from there. And you can see in glorious detail what goes on. So here's our dead mouse, and you can see the first thing that happens is that the abdomen starts to distend. And the reason that that happens is because the gut microbes are now proliferating like mad, producing a lot of gas, and that's pumping up the abdomen. And eventually, the pressure within builds to be so great that the body wall ruptures and fluids seep out, and that triggers the spread of microbes breeding like mad all over the dead body. And you can see that visualized here by the spread of fungal mycelium. You see here it comes, starting to consume the dead body. So all these microbes that are in the body and in the surrounding soil and on the surface of the mouse, they too would like to breed on, use these resources on the dead body to breed for themselves. And so that's a serious issue for burying beetles that also want to use those resources. And it's not just microbes that are a competitive threat to beetles. They also face competition from other insects. And that competition can come from other burying beetles who also want to use the dead body to make a nest for their larvae, or it could come from blowflies who are super quick to locate a newly dead body and to lay their eggs on it. And so what I want to tell you about this afternoon is the different ways in which the beetles manage this competition so that they're able to go ahead and breed on the dead body themselves. And you'll be interested to know, as you're sweating away here on the hottest day of the year so far, that temperature is a recurring theme. Changes the way that we behave and interact with each other, and it has a profound effect on these insects too. So let's start off by thinking about how the beetle manages competition with microbes. And for this part of the talk, we're going to focus on work that was carried out by this brilliant postdoc in my lab, Anna Duarte. And the first thing that we're going to think about in how beetles manage competition with microbes is the way in which they convert the dead body into a nest for their larvae. And so to do that, we're going to look at some more time-lapse footage. It's in black and white, so don't worry, there's minimal gore. And we're going to look at about 2 days' work condensed into 30 seconds as a pair of burying beetles converts this mouse into a nest for their larvae. Okay, so what you can see is the beetles move the mouse around a bit, and then they home in on the abdomen, they make an incision, and they pull out the guts, and they consume them. And then you can also start to see this white stuff accumulating on the surface of the soil. That's the fur, the hair of the mouse that they've scissored off. So eventually the mouse is completely denuded, and at this point, together, they begin to roll that flesh into a ball. And if we'd given them enough soil, it would be below ground by now. And so you can see that by the end of this 2 days of work, we have this beautiful sphere of flesh glinting slightly in the light because of all the exudates that the beetles have added to it from their mouth and from their anus. So here's a close-up of the beetle's abdomen. These are the exudates that we're talking about, this brown stuff coming out of their rear end. And so there's one of 3 important components that go on during carcass nest preparation, which are vital for defending the carrion from rivals, and in particular for managing microbial competition. It's very important that they take the guts out because that prevents rupture and so slows down the process of microbial colonization of the carcass. It's very important that they bury the carcass because that hides it from rivals. And this third line of defense against rivals are the exudates. And they contain lysozymes and fungicides, which lyse bacterial and fungal cells, which might be rivals for the resources on the carcass. And we found through our work that they particularly target pathogenic bacteria. So they take out bacteria that could be potentially harmful either to the adult beetles or to the larvae. But these exudates also contain bacteria. And that's because when the beetles eat the mouse's gut, they eat the mouse's gut microbiome as well. And so that microbiome passes all the way through the beetle and out through its anus and back on to the surface of the carcass. So the beetles are working very hard here to restructure the microbiome on the carcass. They take out the things that could be potentially harmful for them, but they're also adding back in microbes that they have recycled from the carrion itself. And so the obvious question is, well, why are they bothering to do this? Is it in any way beneficial to them? And we tested that idea by using germ-free mice. So germ-free mice are an invention for medical science where people have discovered it's possible, it's labor intensive, but it is possible to produce mice that have absolutely no microbes on them anywhere, either inside or outside. So obviously that's a fantastic tool for us in our experimental work. And so we allow beetles to breed either on germ-free mice or normal germ-ridden mice. And we cut the mice in half, so we had a head end and a tail end, and we gave beetles bits to breed on in regular soil, and then we compared their performance by seeing how heavy the larvae were by the time they dispersed away from the carcass. And the results of that experiment are shown here. So here are our two treatments. These are beetle larvae that were bred on control mice, so covered in microbes, or germ-free mice, completely depleted from microbes. And here's a measure of how heavy they were by the time they dispersed away from the carrion nest. And the main message to take from this is that the beetle larvae perform much better when there were lots of microbes present than when they were removed. And so that suggests that the beetles really do need those mouse's microbes to be able to breed successfully, because otherwise their larvae will be very weedy indeed. Now, we don't know why the microbes are so beneficial to the larvae. It's possible that they somehow trigger parental care, so they make parents express better care. That's one option. Or it could be that the microbes themselves are directly beneficial to the larvae. They carry out some service, like a digestion service, that helps the larvae grow to be bigger and fatter. We don't know which of those two possibilities is more likely at the moment. We need to do more work to find that out. But we do know that the microbes need to be there for the beetles to perform effectively when they reproduce. So this tells us then how beetles start to manage competition from microbes. What we've seen is that the beetles can modify the microbial community on the nest by taking out threats to them that are potentially pathogenic, but also by adding back in microbes that seem to be beneficial. And we've seen in particular that the beetles need the mouse's gut microbes to be able to breed successfully. So that's all I'm going to say about microbes. Let's move on and think about how they manage competition with blowflies. And the work that we're going to think about for this part of the talk was carried out by a PhD student called Seung-Jun Sun. Now, if you catch a wild bearing beetle, then it's very likely that you'll find one that's covered in mites, like this. And they are a particular kind of mite. They're specialist on bearing beetles, and they're called Poikilocheiros carabi. And the reason that these mites are associated with the beetle is because they too like to breed on a dead body. But they've got a major problem, which is that they can't fly. So they use the bearing beetle as a personal private jet to travel from one breeding opportunity to the next. And they ride on the beetle as a friendly, benign passenger, so they just cling on to the beetle's hairs. You can see they've got this specialized chelicerae for holding on tightly while the beetle is in flight. And then the moment that the beetle finds a dead body to breed on, the mites hop off and they breed alongside the beetle. So they travel as sexually immature deuternymphs. And then when they get to the carrion, they immediately molt into adults and they start breeding. And their life cycle is timed perfectly to match the duration of beetle parental care. So that means that when the beetles have completed parental care and they're heading off in search of a new piece of carrion to breed on, the mites, the next generation of mites, is ready to hop on to the adults and fly off with them. And there's usually about 10 or 20 mites traveling on the beetle between breeding opportunities. So it's really obvious what the mites are getting out of this arrangement, because they get these free flights between feeding and breeding opportunities. But the big question is, what on earth are the beetles getting out of this in exchange? And so that's what Sun's PhD thesis aimed to try and find out. Now, it's long been speculated that mites help beetles out and help specifically in the competition with blowflies because they eat any blowfly eggs that they find on the carrion. But nobody had really put that idea to the test in field conditions. So that's what Sun did at one of our study sites near Cambridge, this time at Madingley Woods. And so he set up pairs of beetles to breed on a dead mouse at each of these different locations marked with a yellow dot, and he used them again and again through the beetles' breeding season. And before he set them up to breed, he took away all of the naturally occurring mites on the beetle, and then he added back in to the pair either no mites or 10 mites or 20 mites. And then he allowed the pairs to breed, and they say they were exposed to natural attack by blowflies. And at each breeding event, he measured the air temperature very carefully. And then at the end of reproduction, he exhumed the dead body and he measured the reproductive success of the beetles and the blowflies by counting the number of larvae produced by each species. And the results of that experiment are shown here. So first of all, what we're going to look at here is the reproductive success of bearing beetles and blowflies across a temperature, the same temperature gradient. And this time we're looking at conditions where no mites are present at all. And what you can see is that beetles have peak reproductive success at this intermediate temperature here, but they do rather badly at lower and higher temperatures, because here they're leaving many fewer larvae. But we see the exact opposite pattern for blowflies. So at intermediate temperatures, they have fewest larvae, but they perform really rather well at lower and higher temperatures. So this suggests that the beetles and the blowflies are in competition. At intermediate temperatures, the beetles have the upper hand, but at lower and higher temperatures, it's the blowflies that have the upper hand. And this means that the beetle needs help specifically at these lower and higher temperatures to cope with the threat of blowflies, and that's where the mites come in. And you can see that from these experimental data. So here's our default condition with no mites present, and now we're looking at beetle reproductive success when there are 10 mites present and 20 mites present. And you can see that with 10 mites present, their reproductive success is greater at lower temperatures, and with 20 mites present, their reproductive success is greater at higher temperatures too. So this suggests to us that the mites are part-time allies for the beetle in seeing off rival blowflies. And that alliance is particularly important to the beetle at lower and higher temperatures. And you can imagine that that alliance is only going to get more important for beetles in the future as our climate continues to change. Now, it turns out there is a side effect of beetles teaming up with mites, which we were a bit surprised by. And the side effect is the effect of the mites as they walk across the carrion and they graze on it. It's the effect that they have on bacterial diversity on the surface of the carrion nest. And so what we're looking at here are 6 dead mice during the breeding event sampled either when mites were not present or when they were. And each of these bars, so each one of these bars represents a different mouse nest, and each of the colours within the bars represents a different bacterial order. And that we detected that using DNA sequencing. And what you can see is, I hope you can see intuitively that when mites are present, the bars are more colourful, more diverse range of colours than when mites are absent. And if you remain to be persuaded that that's the case, or perhaps are colourblind, then here's more quantitative evidence to support that conclusion. Different measures of bacterial species richness in the absence and presence of mites. And you can see that when the mites are present, there's much more bacterial diversity on the surface of the carcass than when they are absent. Now, we don't know yet whether this is beneficial for beetles, but what we do know from studies of gut microbiomes is that a healthier gut microbiome is a more diverse one. And that's because it's very resistant to invasion by pathogenic bacteria. Because if you have a really wide range of bacteria in a microbiome, they're filling every ecological niche. And so it's much harder for a pathogenic microbe to come in and gain a toehold. And so that could be a potential advantage to the beetles here, an accidental byproduct of teaming up with mites, that they've got this additional layer of assistance in managing competition with pathogenic microbes. But that's something that we have to test more formally in our future work. So what we've discovered so far then in this part of the talk is that the beetles manage the competition with blowflies by carrying mites who act as allies at low and high temperatures. And they're also accidental allies potentially in dealing with the threat from pathogenic microbes, although that remains to be discovered. And so in the final part of the talk, I just want to tell you briefly about how beetles manage competition with other bearing beetles, their own kind. And once again, this is work carried out by Seun Jun Sun, and once again, it involves an alliance with mites, although in a very different kind of way to the example that we've just heard about. And the kind of competition that we're going to think about now shows itself when beetles first find a dead body. So on each small mouse, there's really only enough resources to support one family of bearing beetles. So if several beetles turn up at a dead mouse, then they have the most almighty fight to determine who gets to own the mouse and have their family breed on it. And the winner of the fights takes exclusive ownership of the dead body and makes it into their own. And often, the bigger the beetle is, the more likely it is to win these fights. But it turns out that beetles can get a kind of superpower in helping them with these fights, and the superpower comes from mites. And it shows itself by just making them more competitive. And you can see that from the experimental data shown here. So if we pit beetles against each other, and one has mites and one does not, the one with the mites is much more likely to win the contest. So the question is, why? What on earth's going on here? How are the mites helping out? And it turns out that the superpower that the mites confer is that they allow beetles to maintain a high body temperature after exercising. So the mites are like a giant fur coat. So when the beetles carrying mites exercise, they stay warm for longer. So it's a bit like us exercising in a thick coat. When you're running around in the cold, you can stay warmer for much longer in that kind of situation. And we tested this idea formally by building a treadmill for mites and then allowing them to exercise on it. And we had 3 different kinds of beetles in this experiment. So we had beetles that were carrying a load of mites, beetles that had no mites at all, and that beetles that were carrying a load that was equivalent to the weight of the mites, but was in fact just a piece of blue tack. And so we allowed these different kinds of beetles to exercise on our treadmill that we built for them. We can see that happening right here. And we tested small beetles and large beetles, and we measured their temperature immediately after they had exercised, and then after they'd spent some time resting afterwards. And we did that using a thermal camera, which you can see here. Oh, hang on. Where have we gone? There we go. Here's an image from the thermal camera. And so the gradient of colour here allows us to infer beetle temperature, because the whiter the colour is, the hotter it has become. And so here are the kinds of results that we got from that treadmill setup. So here we have the duration of exercise on this axis here and the temperature increase in the beetle for small beetles that were walking on the treadmill and large beetles. And these different coloured lines correspond to those three different treatments. So the black line is a beetle without any mites, the red line is a beetle carrying the blue tack weight, and the orange line is the beetle carrying mites. And so there are two things to take from this slide. The first is that there's no difference between these three treatments for large beetles, so they just all heat up when they exercise, irrespective of whether they're carrying any kind of load or not. But for small beetles, that load makes a big difference, and they increase more in temperature when they're carrying a load than when they're not. But the very interesting part of these experimental data are here on this next slide, because this is what happens to body temperature when the beetles stop exercising. And again, we've got data for small beetles here on the left and large beetles on the right. And so you can see it doesn't matter which treatment the large beetles are in, they're all very good at retaining a high body temperature when they stop exercising. But that's not the case for small beetles. The small beetles rapidly lose body temperature unless they're carrying mites. And then they're able to retain a high body temperature in the same way as a large beetle is. And this is all it takes to make the difference in their competitive prowess. So if we just take a beetle and we warm it up slightly, we can give it exactly the same competitive advantage that it enjoys by carrying mites. And so the mites are beneficial because they're simply allowing these smaller beetles to retain heat after exercise, and that gives them a competitive edge when they're competing for key resources to breed upon. And so just to wrap up this part of the talk, beetles manage competition with other beetles for the carrier nest by teaming up with mites who improve their fighting prowess by keeping them warm. And so what we've seen then are three different ways in which beetles can manage the competition with microbes and other insects for resources on the dead body. And they each involve forming alliance, but with different types of alliance for different kinds of rivals. And in some cases, these alliances are sensitive to temperature, and it might be that these alliances become even more important in the future in enabling beetles to breed successfully as our climate continues to change. So I'd just like to end by thanking these very generous funding bodies for supporting our research, and this fantastic team of people that have helped out over the years, and you for listening so carefully. Thanks very much.